All right, good morning. And our webinar today is a special topic today with Dr. Duma. And I would just like to do a quick review, just saying, let everybody know that you are on mute. You will be on mute for the webinar. There is a chat box. If you have any questions, enter that into the Q&A, and we'll get to those at the end. So thank you for joining our webinar today with Dr. Duma. Dr. Duma, he has, he's a board certified neurosurgeon, medical director, the Hoag Memorial Presbyterian Hospital Brain Tumor Program. He's got quite a bit of experience with deep brain stimulation for treating Parkinson's. He's also a CEO and founder of Music Heals Project. There's quite an extensive experience here, Dr. Duma. Thank you so much for joining and presenting this topic for us.
Great. Thanks a lot, Jasmine. I appreciate, first of all, I thank you, Jim McAleer, for giving me this opportunity. This is a very exciting topic, I think. And for those of you in the audience, and I hear it's a pretty big audience today, I think it's going to be very exciting for you to hear this because it's a completely new approach to the treatment of Alzheimer's disease. You've all, I'm sure, been following the progress of, or lack thereof, of the various treatments for Parkinson's disease over the many years. And this is really what gave me the impetus to go forth in this direction. Notwithstanding, I am a brain surgeon, so I'm kind of focused on the actual brain itself and not pills. And I'm going to describe that in a few minutes. So I'm going to share my screen and we'll start the show. This is a very new topic for anybody. I mean, if you looked at the original topic, it's injecting stem cells, not IV, not intravenously, but directly into the brain. And I'm going to describe for you how this moved along over the last 8 years. I'm going to basically describe my journey from the get-go, how this all began. And it starts basically as to, what made me do this? What really was in my mind, my thinking when this happened? Well, I told you all I'm a neurosurgeon. The year was 2014, so that's 8 years ago. And I'd see one Alzheimer's patient come in the office, family members, ALS patients, multiple sclerosis, Parkinson's disease, chronic traumatic encephalopathy from head injuries. And we have nothing for these diseases, and we've had nothing for decades. The actual statistics are kind of interesting. If you look at them, between 1995 and 2021, that's a 26-year period, about 900 drugs were approved by the FDA. And of those 900 drugs, only 6 of them were for Alzheimer's disease, which is fascinating. And by the way, not one new novel therapy for Alzheimer's disease had been introduced into the market since 2003. So that's 20 years of a barren pharmaceutical industry for Alzheimer's disease. The famous aducanumab, or Aduhelm, came about in 2021 and was barely approved and is extremely controversial. The European Union chose not to even approve it for their patients because it's so expensive and it doesn't work very well. And in fact, there were some serious side effects that occurred from that drug, brain swelling and bleeding, to name a few. If you're in with all the latest media and press, the same company, Biogen, has introduced lecanemab, which is another antibody which targets, amyloid beta, which we'll talk about later, this September. And my goodness, their stock price went up 5x that very day because it had some effect on actually prolonging the life of an Alzheimer's patient and prolonging their or delaying the progression of the disease. One of the first drugs in the world that's done that, but it only occurred in about 27% of the patients, and it also still has some controversial side effects, including brain swelling and bleeding. So the real problem is, if you, I love this analogy. This is a take-home analogy for you all. What the pharmaceutical industry has been doing for the last 4, 5, 6 years on treating this disease is they are basically taking out the garbage that is being made by the sick brain. So let's think of an Alzheimer's brain as being a broken refrigerator, and it's producing spoiled food. So the food in the refrigerator is spoiling. If you keep taking out the spoiled food and throwing it away, which is what the antibodies are doing, that's not fixing the problem. It's not fixing the refrigerator. Okay, so if you look at what the approach has been, it's been targeting beta amyloid or amyloid beta, the plaques that occur in these Alzheimer's patients, and removing that very effectively. But is that really the problem, as the rest of the refrigerator or the brain is not working? And to me, that is a real problem with the strategy of treating this disease. So there are two major problems with treating Alzheimer's disease. One is, everybody knows about the blood-brain barrier. If you take a pill orally, it gets finally into the bloodstream and then may make it to the brain, but the brain has this famous blood-brain barrier, and you can't really get past it with large molecules, et cetera, various effective, antibodies, stem cells, things like that can't get through the blood-brain barrier IV. It's just very simple. The second problem is Alzheimer's disease is a disease with multiple causes, multiple etiologies. Amyloid beta is just one end product of the cell dysfunction in the neurons of Alzheimer's patients. And I'm not going to go into all this. Is another scientific discussion, all the different ways that Alzheimer's disease manifests itself, but there are so many problems that are going on in the Alzheimer's brain that you can't even begin to think that a single pill targeting one particular target would create the successful treatment that you want. So what could do this? And the answer for me, very simply, was stem cells. And the year, as I mentioned, was 2014, so it's 8 years ago. I said, we have to inject stem cells directly into the brain. We can't inject it IV. It'll get stopped in the lung, and it won't get through the blood-brain barrier. Intraarterial injection would be the same problem and more dangerous. Interestingly, it's extremely safe to inject things into the brain. And just take it from me, I'm a brain surgeon. It is, and I'll explain how we do that in a moment. But I thought the perfect route would be the ventricles. Now, a lot of you don't know it, but our brains are hollow. In fact, most patients that come across my desk, they have no idea that our brains are hollow. It's like a honeydew melon. You cut the melon in half, and there's a hollow area in the middle. These areas are called ventricles. They're filled with fluid that bathes the entire brain. In fact, this fluid is produced by the brain every day, 500 cc's, which is about 2/3 of a bottle of wine, of fluid that looks like crystal clear, pristine spring water. And what it does is it irrigates the brain of all the byproducts of cell function, and cleans out our brain, literally washes our brain. We're being brainwashed on a regular basis. This fluid is always there, and it circulates into every nook and cranny. And not only does it go to every nook and cranny, it goes into actual right next to the neurons, and it can actually deliver things into those neurons. And so if you put a port into that area called the ventricle, it would seep into the brain. Let me show you what the edge of that ventricle looks like. It's got a ciliated epithelium with actual non-tight junctions here. So if something was in the ventricle, it could actually seep through this area. And oh, lo and behold, what is right here? These are all the building blocks of neurons and neural glia, neural progenitor cells, neural stem cells are living in the edge of our ventricles at all times. Everyone in this audience who's attending, we all have stem cells living in the nooks and crannies of our ventricles. So why don't we use them? Why doesn't the Alzheimer's patient use these to replenish a sick brain or an unhealthy hippocampus, for example? Well, I think we've outgrown or out-evolutionized our ability to do this. And that is up for debate, whether we can actually stimulate these stem cells that are sitting here, that's the blue little things here, to actually get active again and repair and seek and repair the damaged tissue. And that's really the hypothesis of using stem cells. You know who uses stem cells? The rat. The rat uses stem cells all the time, because if a rat can't smell, or as time goes on, they get older and their sense of smell starts fading, the rat actually has a way of reproducing its own neurons for the sense of smell, the olfactory bulb. And this is a well-known process that the scientists have discovered this. And where do these cells come from? That subventricular zone area with stem cells in it. The stem cells propagate, they create new neurons, and they repair the rat's sense of smell. To date, we really don't know whether we have anything like that in the human, but it's my theory that we can probably stimulate this type of thing. So my thought on this matter is that if we introduced stem cells into the ventricles or the hollow cavities of the brain, they could literally percolate into the brain itself and actually start stimulating some stem cell activity. So we did our first work on, of course, the rat. So this is a rat being operated on stereotactically, where we proved that we could actually get into the ventricle. So this is some dye. This is blue dye that shows the ventricular system of a rat. So you see the rat also has a hollow area in it. And once we're in there, we can inject stem cells. And it's very hard for you to see this or even understand it, but this box up here shows that what's inside this box here. And notice this box is deep in the brain itself, not in the ventricle, but in the actual substance of the brain. And the stem cells made it through the ventricles of the rat and into the brain itself. Many other scientists have shown this. So the brain ventricle barrier, which there is one when you're a baby in utero to protect the fetus even more, opens up when we're adults, and we can get things into the from the ventricles into the brain that we couldn't do when you were, 1 or 2 months old. It's kind of interesting that Mother Nature has allowed us to do that. So with this in mind, we took our studies privately through an institutional review board, and we got approval from an IRB to treat a number of patients for different diseases. And all of these patients had the type of disease that had no known cure or even any treatments. So the Parkinson's disease patients, for example, we have lots of drugs for Parkinson's disease, but these were Parkinson's disease variants. These are patients that we call Parkinson's plus. We have not one drug for these diseases. ALS, you all know about that. Chronic traumatic encephalopathy, we have nothing for that. MS, we have a number of drugs, but we targeted the patients with progressive MS, some stroke patients, and of course, patients with Alzheimer's disease. And the study, as I said, which was done privately, was to inject their own stem cells from their own abdominal fat every couple of months through a port that gets into the brain. And now here's the port. So the patients would have an implanted port here. It sounds a little horrible to some people that we were drilling a hole in the head and putting a tube in there, but that really is the only way to get the cells into the ventricles of the brain. Interestingly, this is a 15-minute operation, has no pain involved, and the patient now has this port sitting under their scalp, and I'll show you how that works in a minute. So the patient underwent a placement of this reservoir called an Omeyer reservoir, and that was done before we did a liposuction and gave them cells. A lot of you in the audience may know someone with cancer, and they have something called a portacath, which is in a large vein under the collarbone. These portacaths are used to give chemotherapy so that you don't blow peripheral veins in the arm, and makes it a lot easier to get chemotherapy. But this is basically a portacath for the brain. In fact, it was invented for chemotherapy also, and I've been using it as long as I've been in neurosurgery, for about 35 years. It was invented 50 years ago. So it's been around. Every neurosurgeon knows how to put these in. It takes only about 15 minutes to put in. And this is what the process was in the first phases of my work. A patient underwent a normal liposuction. And it's like a mini liposuction. It's not like a plastic surgeon's liposuction. And then the cells are spun down, and then the spun down product, which is called stromal vascular fraction, contains the stem cells, and then it's injected into the port. And here's a real person showing how it's injected into that port, which is there after it's being sterilized. This is what the solution looks like after it's spun down. And this is the bedside, patient's bedside setup. This is the solution we use to clean the head. This is the butterfly needle we use to introduce the stem cells. And then we wash it with the patient's own spinal fluid afterwards. So patients really tolerated this without any difficulty. This is the concept here, and this is what it looks like close up. There's literally no pain, just a little pinprick when that goes in, no pain to have that injected. And the patient lives with that in their head forever. We wound up doing over 5 years, as I mentioned, in a private study, IRB approved, pre-FDA, 5 years of human clinical trials in already 39 patients, 158 injections in those 39 patients, with about a little more than 5-year follow-up. And for the first time, interestingly, we saw some changes, especially in Alzheimer's patients and in multiple sclerosis, that were astounding. Nothing less than monumental. Of that group of 38 patients, 39 patients, 12 of them had Alzheimer's disease. And we really were doing this study just to see if it was safe. And the safety was superb. We had the 10% rate of some headaches, which we fixed with steroids. And out of the 158 injections, we had very few, we had actually no serious adverse events. We had adverse events, but no serious adverse events beyond, say, headache. We had one patient that had meningitis, but they had dental work done the time before, and that was out of 158 injections, which makes a less than 1% rate of meningitis. So it was extremely safe, especially compared to some of the results we're seeing with aducanumab and the new drug. So we treated 12 patients over 5 years. Now, this is an Alzheimer's audience, so you all understand this Alzheimer's curve. Okay, this is a mini mental status score. As you know, the highest score is a 30 on the mini mental status exam, and you can follow an Alzheimer's patient once they're past mild cognitive impairment and they're diagnosed with true Alzheimer's disease. The disease will progress over a period of 6 to 8, 7 years, and in this fashion. And unfortunately, there have been no drugs from the FDA that have changed this curve. Supposedly, the new Biogen drug moves this curve out a little bit, but doesn't change the actual shape of the curve. So as the patient progresses, the scores on these mini mental status tests will change, obviously, for the worse with time. There is no drug on the market right now that has ever changed that. And we started looking at some of our patients who had been treated. And for example, we saw this. So this is a patient that had been followed for a long time because her sister had Alzheimer's disease. And so she had been followed by University of Kansas for her mini mental status score well before she was diagnosed with Alzheimer's disease. And indeed, she was diagnosed right about here. And this is her mini mental status score going down. She got down to 15. And then we gave her her first injection of her own stem cells into the ventricles, as I described before. And for the first time ever, we saw leveling off of the mini mental status score. Was this spurious? Was this a one-off? Is this real? So we followed her even farther out. Now, this is a 2-year interval, people. So she was followed here. The red line is where she should be. The blue line is her. And this is for 2 years. She had stabilization, in fact, some slight improvement of her mini mental status score. That's monumental, never been seen before in the history of medicine. And that really excited us. And also her p-tau levels, the phospho-tau levels were going down. The beta amyloid levels were improving significantly. And this was very exciting for us. And then we looked at a number of other patients who could be followed for more than 4 months. And look at their trends. Their trends are pretty similar. All had stabilized. And each one of these marks here is when they got an injection and when they got a mini mental status test. So this was not just a one-off of one of our patients. And by the way, that patient went on for 4 years after injection. I have another patient of ours that went on for 5 years after an injection. Who basically died of his heart disease while after playing golf for all those 5 years, very functional. His wife can attest to that. So we saw all these changes here in this pre-FDA work, and we were very excited. How about the spinal fluid levels? Everybody knows about beta amyloid and phospho-tau. These are the plaque forming and neurofibrillary tangle forming elements that you see in Alzheimer's patients, but we're finding out now that they may not be the cause of Alzheimer's disease. Remember the refrigerator analogy. You got to fix the broken refrigerator. You can't just keep taking out the rotten vegetables or the food. So this test is purely objective because it's the spinal fluid levels of p-tau, which is the end product of cell death. So when you do this test, your score is scored in this little histogram here. And if your spinal fluid levels, this is done with a spinal tap, If your spinal fluid levels of p-tau are out here, you're considered to have Alzheimer's disease. If they're anywhere else, you may have something different, but not Alzheimer's disease. And furthermore, they take into account the level of beta amyloid as well. So beta amyloid is indexed over here. And if you have this low number in this index and this high number here, you sure have Alzheimer's disease. And if you did anything to change that trajectory, it would be monumental. So we looked at one of our patients, and this was on day of their first, this was before their first stem cell injection. This was 4 months later, and this was 8 months later. So you can see that the trajectory of this patient's spinal fluid was improving p-tau levels, which everybody, this has never been seen before in the field of medicine. So this was very exciting to see that the p-tau levels are actually improving in these patients. And again, we did this anecdotally. We were looking just for safety, but we saw these very interesting pearls in certain patients that gave us real hope for this disease. We even saw one patient whose hippocampus, that's the short-term memory area of the brain, we saw that actually grow. And we have a way of measuring the hippocampus. Artificial intelligence computer measures the volume of the patient's hippocampus. That's this olive green area. And that volume actually increased over a period of 5 years in this patient. They usually come in, all the Alzheimer's patients usually come in with a very shrunken, shriveled up hippocampus, which is less than 5th percentile for their ages. And to see any change in that would also be extremely surprising and, again, monumental. We saw this in one patient. So this actually gave us some pause that this really should be investigated. Multiple sclerosis, this is not a talk on MS, but I have to show you this video. This is a young girl who had progressive MS, who we treated also for her problem with our stem cell solution. And this is what she looked like when she first came into our office. She was walking 1-inch steps, if you can see that video. So she had already been to USC. USC has a fantastic MS program, and they are using every new drug on the market in experimentation for this type of patient. And she was on it for 10 years. She took our stem cells, and this is her after 4 months, already walking farther, stronger, bending her ankles. And remember, she has the progressive form of MS, and she walked 10 feet in 1 minute, whereas she started at 4 feet in 1 minute. Now she's 14 months out. Frankly, she should be in a wheelchair by now, and she's walking even stronger, bending her ankles, and far more independent than she was. And the only treatment she had was the stem cells that we put in. She has a port in her head, and we instilled the stem cells through the port every 3 to 4 months. And now she finally was ambulating 25 feet and driving a car. So what happened was she got a phone call from USC, and they said they had a brand new drug they'd like her to try, and she'd have to get off of our stem cells and stop our stem cells in order to do the study. And she'd have to do that for a year. Now, she chose to do that. And so she stopped our stem cells. She went on the study drug. And 1 year later, she came to our office. And unfortunately, this is what she looked like. She can't even take one step after being off the stem cells for 1 year. and she had regressed even worse than when she arrived at us three years prior. To me, this was a very eye-opening event, that the stem cells clearly did something for her. We published our work, and this is in the scientific literature. After about 3 years, I published this in a major journal, peer-reviewed journal, and that put us on the map as the first in the world to do this type of thing. There's been only one other trial done in Korea that used not autologous cells, not cells from the same patient. They used fetal cells, and that didn't work out very well for them, for the immune response that you might imagine occurs. But we have the footprint in the scientific literature right now to say that we are the first who's ever done this. And we made a decision to actually form a company and submit an IND to the FDA. At this time, the FDA was cracking down on stem cells being used without their approval, and I said absolutely fine. So what we did was we brought everything to the FDA. We submitted a formal IND. We formed a company. We had to capitalize it. We needed money. We sold stock in the company to get money to continue this research. And we literally submitted in 2020 our IND to the FDA. So now you're talking almost 2.5 years ago. And what did we submit? We submitted our pharmaceutical, which now, because it's FDA controlled, we can expand it in the laboratory, and we can also supercharge it with a special signaling protein. So no longer was it just plain fat spun down, but now it's expanded cells supercharged with a signaling protein that will remember that those cells, that the building blocks of neurons that are right next to the ventricle, that substance, the signaling protein called Wnt, W-N-T, is what we select our cells for. And we believe that that is the on-off switch to signal other stem cells to get moving. And we call our product CereWnt, like brain, cerebral, and Wnt being the signaling protein. So that's the name of this. And we thought that this would be a no-brainer for the FDA because of all the work I did prior to this. It was, remember, 39 patients, 158 injections over 5 years. But frankly, they didn't care about it at all because it was very different from what we had proposed to use as our pharmaceutical going forward, which is a much more powerful pharmaceutical. So in June of 2021, about a year after we submitted, they came back to us and said, hey, you got to inject into a large mammal. Now, what that means is a large mammal, it could be a monkey, it could be, a pig, and we chose a pig. So everybody, that is a pig. We did the study on 12 pigs down in San Diego. This is me operating on a pig. This is me implanting a reservoir into a pig. We have a video of it, but I'm not going to go into it. And there's our famous pig after surgery with his little head dressing on. When we finished this was a 1-year study of operating on pigs, harvesting the cells afterwards, making sure that their organs were clear, that the cells in the brain didn't get to their liver, their kidneys, their other organs, and it was safe. And indeed, all that proved to be true. We sent it to numerous laboratories in Indiana, in Kansas. And so we submitted all that to the FDA, and lo and behold, this letter is what every researcher wants to see. That is a remove from clinical hold letter that said, if you can read the fine print, we have removed the clinical hold and you may proceed with your study. Obviously, lots of champagne was broken that day, and we had a celebration. It had been a 2.5 year ride with the FDA and about an 8-year total ride with my pre-FDA private work. And it was an exciting day. And that's only a few months ago. So we are now approved for the first time to put stem cells that have been supercharged and expanded in the laboratory into the ventricles of the brain. And we just had to make a decision. Where should we do this first trial of its kind in the world? Where would this be performed? And of course, we chose no other than Hoag Hospital, right in all of your backyards. At the rate we're going, the actual first patient may be injected as early as this January, which is very exciting. That'll be a very nice New Year's present for the scientific world. The budget for this is about 1.4 million just to do 9 patients. Now, the FDA has approved us to do a phase 1 trial. Everybody, a phase 1 trial is a safety trial. We are not really looking for efficacy, but we will be following these patients with MRI scans, spinal fluid tests, and everything to see if there's even a hint of efficacy. But this is purely a 9-patient safety trial. The budget for this is 1.4 million. And obviously this is financed either by buying stock in RBI or naming a donation of that amount of money to RBI or a named donation to the Hoag Neurosciences Foundation. So if anybody has an extra 1.4 million out there in the audience, and I'm sure some of you do, it would be wonderful to have your name on this monumental work. The future, what the future holds is that we are looking to finish the phase 1, that's the safety trial, by the end of 2023. And then we would move into a phase 2 trial, which is an efficacy trial for Alzheimer's disease, starting in early 2024, which really is only a year and a few months away. And, you know, we all in the audience know how fast a year goes these days. So it is a exciting time here in Orange County at Hoag Hospital in particular, where we will be doing the phase 2 as well, hopefully. And the beauty of this phase 2 is we've done a statistical analysis. It only requires 58 patients. So we are actually looking at a smaller trial just because the disease is so difficult to treat and there are no treatments that are effective necessarily in the way we're looking at it. So we only need 58 patients to prove our efficacy. That would start at the beginning of 2024. And interestingly, the same exact cells we can use for other diseases, other neurodegenerative diseases. Of course, we will start a phase 2 for ALS at the same time, given the appropriate amount of money. Phase 2 for traumatic brain injury, phase 2 for Parkinson's disease, finally a stem cell study for Parkinson's disease, and phase 2 study for multiple sclerosis, probably the progressive form. So the beauty of this is that the cells, once they're done with the phase 1 trial, in other words, once we've proven the safety of the cells, we can actually branch off into all of these other diseases and see if there's any impact on them as well. I flew through this presentation. I'm a little early. We'll have plenty of time for some questions. And if you want to reach Regeneration Biomedical, those are the numbers. We'll leave those up for a little bit. You can take a screenshot or a camera shot of those. That would be fine. And then you can reach out to our staff. We are looking for a $1.4 million budget for the phase 1 And then as the phase 2 moves on, we're going to be looking for probably another $3-4 million to move into that phase. Thank you very much. And I look forward to giving you some great results with this very exciting new approach to treating Alzheimer's disease.
Thank you, Dr. Duma. That was wonderful presentation and very interesting information. Um, it's, this kind of studies that Alzheimer's Orange County looks into, providing to our community this cutting edge information and education. So thank you so much. It looks like we do have a couple questions. It looks like we have a raised hand as well. So let me see. First of all, let's go for the first question. What results were observed in those stroke patients?
So we had only one stroke patient in that phase of the study.
Oh,
So we can't really make any statements on that at all. If we did a stroke study, it would involve many more patients than that. So no, it's just, remember, the original intent of that private phase of this study was for safety.
okay. And the second question, what is the cost of having stem cells done?
Okay, well, if you're involved in the clinical trial, it is zero to the patient. So this is all covered by this is why the budget is $1.4 million for 9 patients, because the company, Regeneration Biomedical, will be covering the cost of the trial. And that's how it works. When Pfizer does a trial, when Biogen does a trial, these trials are like $70 million. And they have that money to put toward these trials, and investors put money into their pharmaceutical company for these trials. So we're small, but we're mighty, and we're doing the same. So much smaller numbers, but it's going to be very exciting to see where this goes. So for the patients involved in the study, $0.
Okay. Let's see, Chad Cooper, I'm going to unmute you. Did you have a question?
Yes. Hi. Hey, Dr.
Hi,
Duma.
Chad. Hi, Chad.
I have a question. So let's assume you get through all phases with the FDA. What do the economics look like for Regenerative Biomedical, maybe just on a per procedure basis?
Yeah, that is something that we'd probably want to discuss with my CEO with the company, and is more financial, but I have an answer for you. We, in our estimate of how this would work out, on a per click basis, on a per treatment basis, it would be very similar to the chemotherapy industry. I don't know whether you know how expensive chemotherapy agents are these days, but I mean, one of the more common agents, Keytruda, which you see advertised on TV, and another one, another couple out there, they're usually about $40,000 per treatment. And so that's a lot of money. And when we had our valuation for the actual product, once it's out there in the real market, they took that into account. So, the neurodegenerative industry is a $70 billion industry right now. And there really is no effective drug for it, for a lot of these things. And so that's where we're shooting. But, the pure purpose of this right now is to get something different for Alzheimer's disease at the get-go, and then hopefully it will spill into these other diseases which have absolutely no treatments whatsoever.
Got it. And is there a valuation that you guys are selling shares at this time?
Yeah, what I would do is, this is, purely for the Alzheimer's population here. I'd like you to just take down the number that I showed you at the end, and then we can have you call the office for that.
Okay. Thanks, Doc.
All right.
Thank you. Okay. All right. Let's see. You have another question. Will you accept Alzheimer's patients in the middle stages?
Right. So I didn't go through the inclusion criteria for this particular study. And it's a great question. It's just I wanted to keep it short, but there are a lot of inclusion criteria, and we did that just to make the FDA happy. Okay. First of all, you have to have a PET scan, either an amyloid PET or a FDG PET, that shows that you could have the diagnosis of Alzheimer's disease. Secondly, you need CSF specimens, okay? And thirdly, you have to be what's called a fast stage 4 or 5, okay? If the patient cannot dress themselves, it's kind of a very simple algorithm. If the patient cannot dress themselves anymore and take care of those simple daily tasks, they're probably a fast stage 6 and they would not be included in this. But the beauty of this trial is that we're including the Alzheimer's patients, not MCI, not mild cognitive impairment. You got to realize the new drug that Biogen is touting, it includes the MCI and the very early Alzheimer's patients only. And ours is treating true Alzheimer's patients, fast stage 4 and fast stage 5. And the breakdown, the way I look at it is fast stage 4 is where you start having difficulty paying the bills or figuring out, household tasks, that type of thing. And fast stage 6 is where you're at such a point where you can't dress yourself. So if you're still able to dress yourself and still able to do some of these normal activities of daily living, you're probably at fast stage 5 and you would be included in this trial. And that's really up to your neurologist to decide and the referring neurologist. So if your neurologist is out there and they're interested in this trial, we certainly can look at all of these criteria. I have a nurse practitioner, a PA, a physician assistant, who is specialized in looking at all of these inclusion criteria. And this is, we have the whole process to make sure that you have all the criteria to meet for this trial.
Okay. How does one volunteer to be a patient in the trials for traumatic brain injury?
You're going to have to wait a year and a half. So the good thing for the Alzheimer's patients is we're straight up ready to go, within a month and a half, 2 months. But you're going to have to wait for the Alzheimer's population to get through their little phase 1 trial, and then we can open it up to those other diseases. But traumatic brain injury is a, it's an epidemic. It's out there. It's in all of the concussive sports. It is in PTSD for the armed forces. It's a major problem. So we're looking forward to working with the DoD, Department of Defense. We're looking forward to working with the astronauts. How about that? The astronauts up in space are coming back with mild dementias because they're exposed to cosmic radiation. Okay, it sounds cosmic, but you've heard of X-rays, you've heard of gamma rays. Well, there's cosmic rays which penetrate anything. They penetrate, a 10-foot thick, metal object, and you know that a 10-foot thick metal object is not going to fly in space. So these spacecraft that the astronauts are doing are very vulnerable to cosmic radiation. So that is one of the biggest problems going forward with NASA and the space program. So we've already given talks to NASA on this topic, and they're excited about it. I've met with the NFL, the head of the NFL Hall of Fame, Dave Baker. He met with us for all of his all of his players who have become dementia patients over the years, which is really a sad situation, especially when you see them in person.
Wow, that's incredible information. Thank you.
Yeah, it is.
All right, Bill Tunk, I'm going to unmute you.
Hello.
Hi, Bill. We can hear
Yes,
you.
hi.
Hey, Chris, how you doing?
Doing great. Good to talk to you.
Good talking to you also. I just wanted to give you that there is another tangent angle of going on. I attended the Zoom meeting for people with IPF, as you know.
Mhm.
Dr. Paul Noble, who is the head of the Cedars-Sinai research, what he has come up with is the type 2 stem cells has a deficiency of zinc, decreasing the ability for the cells to regenerate, which would be as far as the lung. And I'm going to just be bold. I'm waiting to get the recording of this meeting. I may try to forward it on to you because of the stem cell link.
Cool. Well, we're,
Okay.
you know, we're open to anything.
Okay, and I may send it to Bill first or whatever.
And
you
the interesting thing about it is that the intellectual property that we have
Yeah.
really we hope locks us into the only ones who will be able to introduce this into the brain,
Okay.
into the ventricles of the brain.
Okay.
So that if these people want to talk to us, there are things called episomes. There's all sorts of other things that research has gone into, and
Yeah.
there's going to be a lot of people saying, "Hey, do you want to inject this into the ventricles of the brain?" And we're going to look into all of those, and
Okay.
we hopefully have the IP tied down for that.
Okay. I appreciate it. Just something I'm interested in, and as
Thanks,
well as
Bill.
all the stuff that you're doing, because I treated a lot of these patients when I was a therapist. So
Yeah, I get it.
yeah. All right. Have a good holiday, buddy.
Thank you. You too.
All right.
Thank you, Bill. Okay, another question. Are injections continuing or is there an endpoint?
Another great question. So if you become a patient in the phase 1 trial, you will move on naturally to the phase 2. Okay. But the phase 2, you'll be getting injected every 3 months. So the phase 1, you get injected one time, and then you wait. We wait to see if you have any ill side effects from it. After you're through with the phase 1, which could be as long as 6 months or even 9 months, then you would be rolled into the phase 2 trial that we call the phase 2A trial. And you would get preference for that to move forward and getting the regular injections every 3 months through the port, which is there still from your phase 1. And we'd carry on with that. And the phase 2 trial will be a placebo-controlled, double-blind controlled study. So you may or may not get the actual placebo, but since the phase 1 patients have already gotten the actual drug, they will probably have their own phase 2A trial to allow them to get the drug itself. So they'll get some preference going forward for being the bold ones who start at phase 1.
Okay, so I guess kind of tacking on that question. So the goal is, I guess, after all these studies are done, would they still have to continue these treatments the rest of their life, or?
Another great question. In my pre-FDA private study, I would give a patient an injection, and about 2.5 months later, the wife or the husband of the patient would give me a call and say, "I think she needs another injection," meaning they got better and then they started getting worse. And that's why it was all built in the get-go. That's the other thing, people go to Russia or they go to Mexico or they go to China and they get a single injection of, reindeer antler juice, or they get a single injection of stem cells. That cannot work. The stem cells, in my opinion, have to be constantly reminded to keep on propagating and to keep on fixing. Now, in the phase 2 trial, we'll be giving over 2 years an injection every 3 months. So what is that? 8 injections over 2 years. But as time goes on, we will probably have another trial moving those out. Maybe you need 4 injections in the first year, 2 injections in this next year, and then maybe only 1 every year thereafter. So that'll all pan out with time.
Okay.
But for the time being, know that it'll be every 3 months.
Okay. What feedback have you received from the scientific neurology field to your professionally published article?
Actually, the feedback for the article has been great. And in fact, you could probably look that up on the internet because the feedback is usually on there. The fact that we're moving into the FDA realm gives us complete credibility and credentials now. So whereas we did this with IRB approval in a private setting before all this, if we stayed in that realm, I think things would be very bad because they're not overseen by the FDA. And as much as people have problems with the FDA, they've been really wonderful to us. When we submitted our IND 2.5 years ago, they sent us back a 16-page, single-spaced document enumerated of the things they wanted us to change about the protocol or do or show or whatever. 16 pages, single-spaced. I mean, that's a lot of work, and it's free. We don't pay for that. It's submitted, but to the FDA. And they came back with that for us. And that's what took us about a year to answer all those 16 pages, single spaced. So that was amazing in and of itself. And then they threw the pig work at us. And I also don't blame them for that either, because we were not in the pre-FDA phase giving the same drug as we were asking the FDA to approve. So the drug that we gave the pigs is exactly the drug that's going to go in the humans for the phase 1. So it's all been very well received, and especially since we're now, I mean, if we're cleared by the FDA, then that is a big green light. And you just ask any researcher, it's the holy grail.
Thank you. Yeah. Next question. How will this benefit Parkinson's patients, and will they be able to participate in any future trials? How far out would this be?
Okay, just like the traumatic encephalopathy and ALS, it'll be probably closer to 1.5 years away. But like I said, 1.5 years goes very quickly, but it doesn't go quickly if you have that disease, especially ALS. So we're moving ahead. We could actually consider, after the phase 1 trial, expanded access use. It's called expanded access protocol. That means that if you have ALS and you're dying of ALS, and we've already proved that the drug is safe, we may get FDA approval to give you that drug if you have ALS. So that's without starting a phase 2 trial. So there is that potential for people going forward, and the FDA is accepting that more and more these days. So to answer the question about Parkinson's disease, what do I expect? Well, we expect the stem cells to seek and repair the damaged substantia nigra and start making dopamine again.
Okay. So how was it determined pigs had Alzheimer's?
These were not pigs treated for Alzheimer's. Okay, so these were pigs treated for safety. So all the pig study was to inject the cells, see if they got infected, see if the cells went into the heart, the liver, the lungs, see if they migrated into the body somehow, and created some form of havoc, or caused hydrocephalus in the pigs, for example. These are being injected into the ventricles, and you could get some hydrocephalus from them from clumping of the cells, but the cells are so small they sift right through the brain. So we had no problems with that, and the laboratory studies that came from that were clean, and that's why the FDA gave us the green light for the release of clinical hold.
Okay. All right. Great. Thank you so much. I think that's really concluding our webinar. Thank you, Dr. Duma, sharing your time and expertise with us. We will be sending out a survey in about 24 hours after the webinar ends. And also you can always visit our website, alzoc.org, to learn about our education programs, our webinars. All those are free, and we have recordings on there as well from past webinars. Thank you so much, Dr. Duma. Thank you again for joining our webinar, all to our attendees. If you have any other further questions, you can always definitely reach out to Dr. Duma at his office or his website. If you have any questions for us, you can always call our helpline, or else go to alzoc.org. Thank you so much. Have a great day.
Thanks,
Happy
everybody.
holidays.
Happy holidays to all of you, and let's look forward to a great new year with some new treatment options.